So hope you all had a nice Labor Day break amid the ash fall and the snow. Where we're headed now is we're going to start talking about DNA structure. On Friday, we finished up the story about axoplasmic transport and moving of vesicles along microtubules and moving of vesicles along actin substrates. Today, we're going to move into something entirely different. We're going to start talking about the DNA molecule, and it's really, really hard to overemphasize the importance of the DNA. DNA is the fundamental molecule of life. And in order to be the fundamental molecule of life, it has to obey certain rules. It has to have certain characteristics. Life in the planet as we know it, we define as being able to make copies of itself. So if DNA is going to be the fundamental molecule of life, it needs to be able to make copies of itself, and certainly we'll see today that it does that. But in addition to making faithful copies of itself, it needs to be able to change over time. Because if DNA is the instruction set for all the proteins in a cell, that's going to then provide the instruction set for the whole organism. And so for the organism to change evolutionarily through generation upon generation upon generation, there needs to be change in the DNA. So not only does it need to be able to replicate faithfully almost all the time, it needs to be able to change and make mistakes over time so we can in fact have evolution. And we'll see that that's true for DNA, and much of the DNA function derives from its structure and ability to replicate. It's a remarkably simple molecule. It's composed of a 5-carbon sugar called deoxyribose and 4 nitrogenous bases. Now, it was a long time before they could figure out how these things were put together in a structure, and it was in the early 1950s that Watson and Crick came up with the fundamental structure of DNA called the double helix. And we'll spend a little bit of time talking about how they got to that point. Now, when we talk about the molecule, we're going to see that the deoxyribose provides the molecule for which we can make the backbone. And then each deoxyribose molecule will have covalently linked to it one of 4 nitrogenous bases: adenine, guanine, cytosine, or thymine. Now, I've paired these up here in a particular way I want you to hold on to. Adenine and guanine are purines. That's a general overall view of them. And we'll see in their structure, they're larger than the smaller cytosines and thymines, which are called pyrimidines. But when it all comes down to it, we're going to see that adenine must base pair with thymine and cytosine with guanine. And we'll talk a little bit about why we're restricted to that kind of base pairing. And it has very important implications for the molecular structure of DNA. We're going to see that the molecule, which is composed of these 5 simple characteristics, the 4 bases in the deoxyribose, is composed of 2 strands. Both strands have a backbone made from deoxyribose that's been polymerized by enzymes into this great long chain. Each deoxyribose molecule is covalently linked to its neighbor by something known as a phosphodiester bond. And the phosphodiester bond arises from the fact that we'll use a triphosphate for each of these bases, and that triphosphate will have the energy to provide the covalent linkage. And we'll talk about that. We'll see then that we make these 2 backbones, both made of deoxyribose linked by phosphodiester bonds. And then we'll see that the 2 strands are held together, that we make the strands, and these strands are polymerized from deoxyribose, so those are all covalent linkages. But the 2 strands are held together by hydrogen bonding. The hydrogen bonding is between the bases, between adenine and thymine, cytosine and guanine. It's the only way we can match them up, and we'll see there's steric reasons for that. Then the next idea comes over here. And that is, even though we have 2 strands, we're restricted in the way we put them together. And this is another one of the important insights that Watson and Crick had. And that is the 2 strands are organized in opposite directions. They're said to be anti-parallel. Now, I'll try to explain to you how we suss out the idea of the organization of these molecules, and it derives from the organization of the sugars. When we lay these out, we'll see that the sugars are organized either 3' to 5' in one strand, or in the opposite direction, 5' to 3' in the other strand. And we'll see, by drawing the sugars, we'll see how that can work out. When DNA replicates, the replication of DNA is semi-conservative. And there's a meaning to that term. What it means by semi-conservative is that one strand is the template strand. And that now provides the template from which we make a new strand. So the two old strands of the DNA molecule provide a template for new strands. So we have one old strand and one new strand, and we'll talk about how this gets done. For this replication to occur, we need a handful of enzymes. Now, there are more that I'm going to list here. There are a whole series of them, but there are a few of them I want to point out to you as being the most important. The first one is called helicase. And again, helicase, as the name implies, unwinds the helix. It does two basic functions. It unwinds the two strands, and in addition to unwinding the two strands, it breaks the hydrogen bonding between the complementary bases. This separates the two strands, allowing for this enzyme, DNA polymerase, to come on and read the template strand. It will read the template strand, making the appropriate phosphodiester linkages between the sugars, while at the same time aligning the complementary bases one to the next. Adenine to thymine, cytosine, adenine to thymine, and cytosine to guanine. Now, one of the interesting things about the way we have to replicate this molecule, the way we have to make copies of it, is that this DNA polymerase can only read the template strand in one direction. Always reads 3' to 5'. Because it reads only 3' to 5', but yet the two strands are organized in an anti-parallel fashion. This means, this leads to the idea of a leading strand and a lagging strand. And the leading strand, theoretically, it doesn't occur in biology this way, but theoretically, on the leading strand, the DNA polymerase can read continuously and make the complementary strand without interruption. But that's not true in the lagging strand. What's happening in the lagging strand is we separate the two strands, and now, because of the complementary nature of the bases and because of the anti-parallel organization of the two strands, that means a DNA polymerase can only read the other strand going in the other direction. So this means we get a lagging strand where this strand is read and synthesized in fits and starts. And we'll see why that is. Those of you who have heard of Okazaki fragments will recognize this is important to the idea of the lagging strand. So we'll walk our way through that and try to generate a molecule of DNA. As we get started here, though, I wanted to emphasize something about nucleic acids. Nucleic acids, for our purposes in here, are going to be in two broad categories. One is ribonucleic acid, and we'll talk more about this tomorrow, and the other is deoxyribonucleic acid. For today's discussion, what I want to start with is to contrast and compare this sugar structure of ribonucleic acid and deoxyribonucleic acid. Whereas the backbone in deoxyribonucleic acid is going to be based on deoxyribose, the backbone in ribonucleic acid is going to be based on the sugar ribose. So let's draw this one first. It's an oxygen, and then it's a pentangle of 1, 2, 3, 4, and up here in the flagpole is the fifth carbon. Always on the number one carbon will be a base. For ribonucleic acid, this could be adenine or uracil, guanine, or cytosine. At the number 2 carbon, we have a hydrogen and we have a hydroxyl. At the number 3 carbon, we have a hydrogen and we have a hydroxyl. On the 4th carbon, we have a hydrogen, and 5 carbon, we have a hydroxyl, and a hydrogen, and a hydrogen. Now, don't memorize the structure. All I'll ask you to recognize is that this is ribose. Know that it's a 5-carbon sugar. 1, 2, 3, 4, 5. And compare it here to deoxyribose. Deoxyribose is again a 5-carbon sugar, so we can start out with the same general structure. We can number the carbons, 2, 3, 4. Here's the one on the flagpole is 5. Number 1 always has a base. In deoxyribose, it could be adenine, thymine, cytosine, or guanine. Now, the number 2 carbon is where we're going to see a difference. On the number 2 carbon, there's a hydrogen and a hydrogen. Hence the name, deoxyribose, where ribose has this hydroxyl, the oxygen at the number 2 carbon. That hydroxyl has been replaced by hydrogen in deoxyribose. This will have important implications for the way the helix can form, because this bulky oxygen down here on the hydroxyl prevents the formation of a nice, neat spiral, whereas the hydrogen, because there's not this bulky oxygen, because of this, we can throw this molecule into a spiral. And we'll talk a little bit about how that happens. The number 3 carbon still has a hydrogen, and it has a hydroxyl. The number 4 carbon has a hydrogen. The number 5 carbon has a hydrogen and a hydrogen and a hydroxyl. So the important idea for both of these molecules is that we only have this subtle difference in the number 2 carbon, which implies real structural differences for the DNA molecule and the RNA molecule. But having said that, there's another piece I want to show here on both of these that's important. And that is, normally when we start making these molecules in biology, we just don't use an ordinary 5-carbon sugar. We start out with a modification, where there's the oxygen, there's the 5-carbon, 1, 2, 3, 4, and up here is the 5th carbon. We'll have the base here. But this number 5 carbon here now is going to be modified so that rather than having hydroxyl, it has an oxygen and a phosphorus, and an oxygen and a phosphorus, and an oxygen and a phosphorus. These are double bonded oxygens with an O negative, double bonded oxygen, O negative, double bonded oxygen, O negative, and a hydrogen. This is said to be a triphosphate. This will be true for all 3 of the bases, all 4 of the bases rather, that we're going to put on here. It could be adenine, cytosine, guanine, or thymine. Another one that we'll see commonly, and again, I want you to notice, emphasize here, this is deoxyribose, so it's a hydrogen and a hydrogen. But over here, I'm going to change this a little bit. Here's a number, here's the 5-carbon sugar. This is ribose, so this is a hydrogen and a hydroxyl. Out here, the number 1 carbon is a base. At the number 3 carbon is hydroxyl and a hydrogen. At the number 4 carbon is a hydrogen. And the number 5 carbon here is again a hydrogen, and now O, P, O, P, O, P. 3 phosphates, double bonded oxygen, O negative, double bonded oxygen, O negative, double bonded oxygen, O negative, and it'll be a hydrogen out here that I can't quite fit in. Now, if this base were an adenine, this would be adenosine triphosphate. Adenosine triphosphate is based on ribose. But we could also have a guanosine triphosphate or a guanosine triphosphate, uracil triphosphate, or cytosine triphosphate. Just as we can have all of those bases over here for deoxyribose, but we're limited here for thymine, cytosine, guanine, and adenine. So we'll get into those subtle differences between the thymine and uracil later on, but recognize that we have the 4 bases that we can put out on the number 1 carbon, the number 5 carbon, we can put these phosphates on, whether we're talking about a deoxyribose molecule or a ribose molecule. So any questions up to there?
So somebody wants to know, so DNA has a triphosphate. Is that what you're saying?
Okay, so the question here is, does DNA have a triphosphate? And once we make the molecule of DNA, we're going to lose these triphosphates, except for the number 5 carbon. And we're going to use the energy in these triphosphates to make a phosphodiester bond, which is going to form the polymer between adjacent deoxyriboses in deoxyribonucleic acid and between adjacent riboses in ribonucleic acid. And I'm going to show you in a minute how we're going to use the energy here to make that polymer. Anything else? Okay. Well, let's go to the right-hand panel and then begin generating, sort of an idea of generating a polymer of deoxyribose. And this effectively is the question that was just asked. And we're going to start here with the 5-carbon sugar. Again, we can number them. This is going to be a base. This is 1, 2, 3, 4, and the number 5 carbon up here. We'll start here. I'll put these in orange. With the oxygen to phosphorus, to oxygen to phosphorus, to oxygen to phosphorus. I'm not going to put in all the other bonds. I just want you to recognize it as a triphosphate. Then we'll see in a moment how this gets done enzymatically, but what I want to do is just simply make the polymer and show you that, again, we have, this is deoxyribose, so these are hydrogens here. Here's our 5-member ring. 1, 2, here's the base. Hydrogen, hydrogen. Now up here, we're going to have a carbon. And we'll put the triphosphate on it. O, P, O, P, O, P. Now remember, and I'll put this one in blue just so we can sort of keep track of it. Out here on this number 3 carbon, there's a hydroxyl. And what we're going to do is we're going to link this number 5 carbon here through an oxygen, a phosphorus, and oxygen to this hydroxyl. To this number 3 carbon. So what we'll do is we'll get rid of this hydroxyl. Or at least we'll get rid of the oxygen. The hydrogen, I mean. And then we're going to take the triphosphate, and we're going to cut off 2 of the phosphates. And then we're going to take this oxygen to this phosphorus to this oxygen to this carbon. So oxygen, phosphorus, oxygen bond. This is said to be a phospho diester bond. So we've taken this first deoxyribose and linked it to this next deoxyribose, but we can do this over and over again. We can extend this for literally millions of these deoxyriboses. And each time we add a new one of these, we can choose whether we want an adenine, a thymine, cytosine, or guanine. But again, here's the number 5 carbon. This is the oxygen, phosphorus, oxygen, phosphorus, phosphorus bond. We're going to take these 2, get rid of them, link this oxygen, again, the oxygen to the phosphorus. And we're going to link the phosphorus then to the remaining oxygen that was on the number 3 carbon. Now, it's not too important here where this oxygen originates, whether it was part of the phosphate or whether it was part of the hydroxyl. I'm not concerned with that. But I want you to know that we go carbon, oxygen, phosphorus, oxygen to the carbon bond. Again, another phosphoester linkage. And I'll do one more. Just to emphasize that each time we do this, we can repeat the pattern. Out here's the base. Here's number 5 carbon with its oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus bond. Again, the oxygen, phosphorus. This is a diphosphate, it goes away. This oxygen to a phosphorus. Now to the oxygen. of the old hydroxyl, and we have another phosphodiester bond. We can do this over and over and over again. We can do it a million times. But regardless of how many times we do it, what I want you to see from this is that this end of the molecule, 1, 2, 3, 4, 5. This is the 5 carbon. So we can say that this end of the molecule is the so-called 5' end. And then the other end of the molecule, because remember down here, if we would go these 5 deoxyriboses, or if we would go 5 million, we would still down here have a hydroxyl and a hydrogen, and a hydrogen. We can number the carbons, 1, 2, 3. So now this is the 3' end. This gives us a kind of description of the molecule, an endedness to the molecule. Just as we saw in proteins, we had amino terminus and a carboxy terminus. Here in this polymer, made from deoxyribose, we have a 5' end and a 3' end. And we can string this out for miles and miles. So are there any questions up to there? But one of the things that Watson and Crick found is that first they discovered that these bases had to be the inside of the helix. And secondly, they found that the two strands were organized in an anti-parallel fashion. All of this occurred from looking at Rosalind Franklin's diffraction data. Rosalind Franklin was a student of a guy named Maurice Wilkins. And Maurice Wilkins and Rosalind Franklin were working in London at King's College. And Watson and Crick were working at Cambridge. And they were sort of competing at this time. And when Watson went to watch a seminar, he recognized a particular picture. And he thought he understood the X-ray diffraction data that Rosalind Franklin had presented at her seminar. But he misremembered it somehow. And so he went back and he and Crick talked about it, and things didn't seem right. And one of the things they thought was going to happen is they were going to put the bases all on the outside. And that was the common idea, because the bases were thought to be important in the hereditary model of the gene. Then the important parts ought to be on the outside. Turned out that was wrong. And it wasn't until they studied a particular picture that Rosalind Franklin had taken and given to Maurice Wilkins, and then Maurice Wilkins gave to Watson, that they began to recognize that there's a diffraction pattern which showed a double helix. And that was the insight that, oh, these bases have to be inside. That's the only way we can make this work. And what's more, is the only way we can make it work is to turn this structure, 5' to 3', around so that over here we have the bases organized in a different way. And I always have trouble doing this upside down, so be patient with me. Here's an oxygen. It's the number 1 carbon, 2 carbon, 3 carbon, 4 carbon, and 5 carbon. Here's the number 1 carbon, so this has a base on it. Now this base can be complementary to this base. So let's give them names. Let's say this is an adenine. And this is an adenine, it's going to base pair. This has to base pair with thymine. But the pattern we saw over here still exists. Out here in the number 5 carbon, we'll have an oxygen, a phosphorus, an oxygen, a phosphorus, an oxygen and a phosphorus bond. We can again get rid of these 2 phosphates and form this phosphodiester linkage. The phosphodiester linkage is going to be the same as we saw on the left-hand side. It's just sort of heading in the opposite direction. And so, let's number the carbons here. 1, 2, 3, 4, and 5. And again, here's our next deoxyribose. 1, 2, 3, 4, and 5. If this base is going to be guanine, then this has to be a cytosine. Remember, the guanine's a purine, so it's purine to pyrimidine, and again, purine to pyrimidine. This same pattern will hold up here. I'm getting a little crowded, but this number 3 carbon will have its hydroxyl. We'll get rid of the oxygen, we'll get rid of the hydrogen. And form this oxygen phosphorus oxygen bond. So this pattern is the same on this side as that. It's a carbon oxygen phosphor oxygen bond. This is a phospho diester bond. And we can repeat this again. So number 1 carbon with a base, the number 2 carbon, the number 3 carbon, it's a little smaller, which will be linked by an oxygen to a phosphorus to an oxygen. And then this is the number 5 carbon, which is again has a carbon, OP. It's the triphosphate, carbon, oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus. And again, we can use this and form one more linkage. So here's our base. 1, 2, 3. The carbon, oxygen, phosphorus to oxygen bond. The phospho diester bond. So what we've done so far is we have laid out 2 strands. We've emphasized that this left-hand strand is oriented 5' to 3'. But you'll notice over here, this is the number 3 carbon. So on this strand, this is the 3' end, and this is the 5' end. So these 2 strands are said to be oriented anti-parallel with respect to one another. Well, that's fine. What we have done so far is we've taken the 2 strands, we've taken the sugars, we've polymerized the sugars using the phosphates, and we have this great long strand. This is the backbone. These are the 2 backbones oriented 5' to 3' and 3' to 5'. But the question then is, what holds the darn molecule together? And what's holding them together are hydrogen bonds here between the cytosine and guanine and the adenine and thymine. And these are said to be hydrogen bonds. Well, all we've done so far is show this in 2 dimensions, but let me see if we can emphasize this a little more with a couple of slides. So the first slide is just to show the phosphodester linkages. And to show how they're all put together. Now this shows more detail than I showed. I got rid of all those extra oxygens. But we can follow through with the same ideas, and this slide shows everything that I put on the board. Here's the number 1 carbon. In this case, they have it connected covalently to a guanine. 1, 2. Here's the number 2 carbon. This is where there's a hydrogen here rather than a hydroxyl. It's deoxyribose. 1, 2. Here's the number 3 carbon that's now linked through an oxygen, a phosphorus, and oxygen bond to the number 5 carbon here of the next deoxyribose. Now the way I drew this number 5 carbon would have a triphosphate on it. The energy in the triphosphate, because when we form this bond, this number 5 carbon has a triphosphate in it, and the energy in that triphosphate is used then, we'll see by an enzyme, to form this carbon, oxygen, phosphorus, oxygen bond between the number 5 carbon of this deoxyribose and the number 3 carbon of this deoxyribose. And we can then polymerize this string on and on and on by simply forming these phosphodiester bonds. The only difference here between what I drew and what they have is they continue to show the double bonded oxygen and the O negative. So again, we've polymerized these deoxyriboses, And they're showing out here in the number 1 carbon the the different bases, adenine, thymine, cytosine, and guanine. But the next thing to think about is how we begin to do the base pairing. And I'm going to show you this in a slide. I'll never ask you to repeat this, but I think it's an important slide because what it shows is the charge in the molecules, in the bases, and how the charges in these bases now can bring these molecules together and get the base pairing. So out here on this nitrogen, we've attached the deoxyribose. This is thymine. So this is a pyrimidine. This is adenine. This is a purine. The 2 purines are adenine and guanine. So if you look at these, you notice they're pretty similar. There are slight differences, but they're pretty darn similar. And then this adenine can now base pair with the thymine, and it base pairs because this double bonded oxygen on the thymine is negatively charged. Whereas the hydrogen on this amine out here on the adenine is positively charged. This hydrogen on this nitrogen of the thymine is positively charged, whereas the nitrogen embedded in this ring because of the electron sharing is negatively charged, and now these opposite charges can pull these molecules together and hold them together by hydrogen bonding. You notice here that they show 2 hydrogen bonds. If we now move down here and talk about the cytosine and guanine base pairing, here's a double bonded oxygen out here on the guanine. Here's a hydrogen on the amine of the cytosine. Again, we have this amine group. The nitrogen in the ring is negatively charged and can bind to the positively charged hydrogen on this nitrogen in this ring. And this positively charged hydrogen on the amine can now attach to the double bonded oxygen, the polar double bonded oxygen here in the cytosine. This is the way these 2 molecules fit together. Watson and Crick at the time knew that more or less there had to be this pattern because of a fellow named Chargaff. Chargaff had done the simple biochemical analysis of DNA from a number of different organisms. And what Chargaff found was that there was always an equal molar ratio of adenine to thymine, cytosine to guanine. So that meant that the only way they could put these together was in some way to pair. You had to have equal amounts of adenine and thymine, cytosine, and guanine in a DNA molecule. So they must be pairing in some way. And the story is that Watson went into the lab one Saturday morning and he started cutting out. He roughly knew the shapes of these molecules, and he started cutting them out as cardboard cutouts. And he sat there at his desk taking these cardboard cutouts and trying to fit them together, knowing what the charge would be on different parts of the molecule. And he found the only way he could get them to fit together was to make the thymine match up with the adenine and the cytosine with the guanine. Then that fit together with Chargaff's experiments that showed equal molar ratios. And now Watson and Crick are showing that the only way you can put these 2 together is have the hydrogen bonding between cytosine and guanine and adenine and thymine. In addition, because of the X-ray diffraction data, they now know that these are facing inward, and they know that they are arranged in helix from the characteristic diffraction data of the sort of cross that you see of X-ray diffraction of a helix. So any questions up to there? Okay, well, let me show this molecule. Highly stylized, but it's worth looking at. In the upper portion here, we see there's the phosphate. This is the 5-carbon sugar. We can count this is the base, so this is number 1 carbon. Here's the number 2 carbon. It only has a hydrogen, so this is deoxyribose. This is an adenine, and just as I showed you in the last image, it's base pairing with a thymine. Now, this is the other strand over here. This is one strand. This is the 5' end of this strand. This is the 3' end of this strand. And we see that it's base pairing here with a thymine, and here are the hydrogen bonds. So this begins to show the antiparallel organization of the 2 strands, and this begins to show how they're thrown into a helix. It appears that the lack of the hydrogen down here at the number 2 carbon provides enough degrees of freedom of movement that we can twist these one around the other. And what's happening is these bases now are facing one another and hydrogen bonding within the middle of the molecule with the deoxyriboses on the outside. In some quarters, this is thought to be because this hides this hydrogen bonding away from water. Basically, this makes it stable and makes it so that it isn't going to be damaged by the water. So the hydrogen bonds are hiding in here away from the water, and the rest of the water is surrounding the deoxyribose, but the hydrogen bonds are not disrupted by the hydrogen bonding in water because they're buried in the double helix. But again, what they're showing here are the sugars and the phosphodiester bonds, and they're showing the base pairing. So any questions on that? Well, if not, what we have to think about now is we have to think about the other fundamental characteristic of DNA that we described early on. And that is that if it's going to be the molecule of life, DNA must be able to replicate itself. It must be able to make copies of itself. So here on this panel, I want to begin thinking about the concept of DNA replication. We're going to see that DNA replication is characteristically semi-conservative. What we mean by semi-conservative is that one strand is the template strand, and I should say this is also the old strand, and one strand is the newly synthesized strand, that's complementary to the old strand. So the long and the short of this is we're going to have two strands of the DNA, and each of those two old strands are going to be templates. And then we're then going to use those two old strands as the templates from which we're going to synthesize a new strand, and those two new strands then will be complementary to the old strand. And let's see how that's going to be done. Now here in the middle panel, I'm going to erase what we have, and I'm going to simplify all this rigmarole that went through before with those phosphodiester bonds, and I'm going to show the phosphodiester bonds as a continuous blue line. So that the continuous blue line is going to represent the backbone of the deoxyribose molecule, the region of the molecule where we have the deoxyriboses, those 5 carbon sugars, linked by phosphodiester bonds. And we're going to simplify that to show a simple blue line. And attached to the blue line will be a series of bases. Now I'm going to splay this out a little bit because I've got to put some more molecules in here. But this is meant to be the phosphodiester linkages between the between the deoxyribose molecules. We know from the description on the right-hand panel that if this is the 3' end and this is the 5' end, then for this strand, this must be the 5' end and this must be the 3' end. And then there are going to be these bases. And I'm just going to pretty much put these in randomly. Adenine, cytosine, guanine, thymine, cytosine, guanine, adenine, thymine, guanine, cytosine, adenine. Now, I'm going to try to show that these are complementary. So this adenine is going to be base paired with a thymine, this cytosine with a guanine, this guanine with a cytosine, thymine, adenine, guanine, cytosine, thymine, thymine, adenine, cytosine, guanine. They do here. A thymine. So that's the complementary bases. These are all being held together by that base pairing, the hydrogen bonding. Now I'm going to put a few more out here because we have to start unraveling this molecule. This could be anything. This could be cytosine, guanine. This is a cytosine. This is a guanine up here. And this is a cytosine here. Now I haven't shown it here because I'm just not that good of an artist, but this will be twisted in that helix. So remember, these two strands are wrapping around one another in that helix that I showed in the last slide. For this molecule to be replicated, so we can take this panel and label it as DNA replication and say that it requires helicase. Helicase is an enzyme. Helicase unwinds the helix and breaks the hydrogen bonds. Now, typically, what I'll do here is I show this molecule as a kind of wedge. I think it's easiest to think of it as a wedge. So I'll draw this in as a kind of an orange wedge. Now, don't get the idea that this is the real shape of the molecule, but conceptually, it's a wedge. And what it's doing is twofold. It's taking the two strands and unwinding them around one another. And as it's unwinding them, it's breaking the hydrogen bonds. So it's separating the two strands. So the first thing that happens is the helicase separates the strands. Then the next enzyme to come along here is it's a protein enzyme. And the next enzyme to come along, and I'll show it in green, I'm just going to show it as a green box, is said to be DNA polymerase. There are a bunch of other enzymes we need in here, but the two main ones I want to tell you about are the helicase, which separates the two strands, and the DNA polymerase, which then synthesizes the new strand, complementary to the old strand. It's a pretty clever molecule, and what it does is it will find the right base pairing. It will find a guanine. For this guanine, it will find a cytosine. For this cytosine, it will find a guanine, and it will now form that phosphodiester linkage. So where the helicase is breaking the hydrogen bonds, the DNA polymerase forms phosphodiester bonds. Remind you that these are covalent linkages between adjacent deoxyribose. Molecules. So this DNA polymerase walks along the DNA strand. The DNA walks 3' to 5'. And as it walks along the strand, said to be the leading strand, what it does is it, at each step, finds the appropriate base. Thymine, guanine, cytosine, and forms those covalent linkages between the phosphates and the number 3 carbon, so that we get the phosphodiester bonds. And this goes on, so that this will then, we'll find an adenine, guanine, cytosine, form the appropriate phosphodiester bonds between each of those, which are each of these bases attached to a deoxyribose. This should be the adenine. So you see, we can now walk along this strand, 3' to 5', and put the appropriate bases in and synthesize a new strand. This shows you that this new strand is complementary to the old strand. The old strand now is the template strand, and this is the newly synthesized strand. So any questions to there? Now, if you've been watching carefully, you'll notice I've thrown a curve. I've told you that DNA polymerase has to walk 3' to 5'. It can only walk in that direction. And yet, here's the other strand running 5' to 3'. Well, the implication for that is that this helicase has to open this strand up by breaking the phosphodiester bonds, and then someplace downstream, a few hundred base pairs probably, there will be another DNA polymerase that comes onto the molecule here. So this green structure is meant to be DNA polymerase. It now can walk in this direction. It will do the same thing. It will find this thymine, base pair it with an adenine, find the guanine with a cytosine, cytosine with the guanine, and so forth, and make the appropriate phosphodiester linkages. Should have done it the other way, sorry. So these now have the appropriate phosphodiester linkages. So there is the phosphate backbone in blue. Here are the bases ACGTA, complementary to TGCAT. But now we're moving in this direction. The problem here is that we have to wait for the helicase to open up the strand before the DNA polymerase can settle on this strand and read it 3' to 5'. Many of you may have heard of Okazaki fragments. The Okazaki fragments are the names of the bits and pieces that Okazaki found when he was trying to sort out the story. When he first started this, Okazaki didn't know that there was only one direction that the DNA polymerase could read. And he found that he would get these great long strands that were perfectly complementary to the old strand. And then he saw all these bits and pieces called the Okazaki fragments. And what was happening is when he would stop the synthesis at some point, he would still have these little bits and pieces of the Okazaki fragments because of the characteristic of the DNA polymerase having to read 3' to 5'. So he was getting these bits and pieces that were being synthesized that were the Okazaki fragments, because they were being synthesized in fits and starts, reading 3' to 5' by this DNA polymerase. This DNA polymerase molecule is the same character. We don't have to have a different type of DNA polymerase. They can be the same kind of molecule. They just are forced to read 3' to 5' in both directions. And so this set is then said to be the lagging strand. Now, if you get down in the weeds of this, if any of you have done some molecular biology, you know that we need some RNA in here. These are called primers. Particularly when we're reading in this direction, we have to put those primers in. That gives a signal for the DNA polymerase to find that site and start reading. We get rid of those RNA bits that are complementary, and then when we get to the end of that, we have to tie it together, so there are ligases that tie it together. But don't worry about that. If you like that kind of stuff, I suggest you go read it in another place and follow the rabbit hole. It's a great place to go. But for now, just recognize that what we're going to end up with is we're going to end up with two newly synthesized strands that are complementary to the old strands. That's semi-conservative replication. And what's more is because of the vagaries of the DNA polymerase having to walk 3' to 5', these are being generated as a leading strand and a lagging strand. So any questions to there? The other point I want to make here, if you notice, if you're following along, you'll see that if this is 3' to 5', because it's anti-parallel, the newly synthesized strand is being synthesized 5' to 3', as is up here, because this is 5' to 3', this newly synthesized strand will be 5' to 3'. And that's how we make these new molecules of DNA. That's DNA replication. What we'll do tomorrow is we'll talk about the structure of RNA and work on how RNA is going to be synthesized from a template strand of DNA. If there are no questions, we can sign off. Okay.